chance, you should come over and not just enjoy the Tim Hortons that we have in that building, but maybe visit some of the other scientists that are there that are doing a lot of good aquatic ecology and salmon research. So today my talk is entitled Changing Climate, Changing Behavior Using Physiological Telemetry and Experimental Biology to Examine a Conservation Crisis in Marine Adult Pacific Salmon. I thought about this title a bit more as I was reforming it after I submitted it, and I thought, well, maybe it's not so much a crisis as much as a challenge. And then I thought a bit more, and I realized, well, maybe it's really not a challenge. Maybe it's just the new norm that we're dealing with. So I apologize if I got you here under false pretenses. You're expecting to hear something catastrophic. And in fact, today you might not hear something like that at all. You can decide whether it's a crisis, a challenge, or just the new normal that we now deal with all the time in salmon management. My presentation today is going to cover three general areas. I'm going to go over some background at the beginning, reviewing for you a bit about the Fraser River and sockeye, in case you're not familiar with that. Talk a bit about the complexity and the conflicts that can arise as a result of the diverse aspects of Fraser sockeye. I will then spend most of my time reviewing this phenomenon of early migrating adults and what I call dead fish swimming. And then lastly, I will wrap up with a review of some of the population-specific thermal tolerance issues that we've revealed and studied over the last few years, and why this is now a new biological reality that we can't ignore any longer, and it prevents, or creates, I should say, quite a challenge now for the management system, understanding these different thermal tolerances that populations have within one river system. Okay, back to the beginning. So you're all familiar with the salmon life cycle, I suspect. I don't want to bore you and review ad nauseum, but the one unique thing about sockeye is after they go from the egg to alvin to fry stage, they're spending time in lakes. Usually, most of them go to lakes, usually for 1 year, perhaps 2, depending on where they live. And then after they smolt, they spend up to 2 years in the ocean before they return to spawn again. Most of these fish that return in the Fraser are 4-year-olds. A few are 5, a few are 3, but most of them are 4. The Fraser is a big river system. It's one of the largest rivers in Canada. It's the 4th largest river solely within Canada. It drains 1/3 of the province. It's the largest producer of salmon. I'm not talking about hatchery production. It's the largest producer of salmon in Canada as a single river system. It's the largest producer of sockeye, and this species is second only to pink salmon in being the most numerically abundant. Sockeye form the most commercially valuable species. And they're also the focus of one of the fastest growing recreational fisheries in the country. And they're a very important component of First Nations culture, economy, and environment. Now this slide's a little bit dated now. It only goes up to 2007, but the trends here are important, and I just want to review a few things. First, you'll notice that there are trends. I think soil production goes up and down. We're looking at each bar is the amount of catch, which is in red, and the amount of escapement, which means fish that were not caught, that escaped the fishery. So the total height of the bar is the total numbers of salmon that would have returned in a given year, in terms of sockeye. And you can see that during the 50s and 60s and 70s, there was a general flat, and then during the 80s and 90s, we had a general increase in some years, and then we've had a general decrease in recent decades. You'll also see that there's these bumps that occur, and the bumps tend to occur every 4 years. And so going backwards 2006, 2002, 98, and so on, every 4 years as you go back, you'll see this higher bump. If we go forward in time, 2006 to 2010, you might remember the very big year. Then we're today, 2014, another big year. We have these natural cycle lines that every 4 years tend to be quite big in the Fraser system, and then we have off-cycle lines where we don't have quite the same return abundance. This is a natural phenomenon. We also see ocean conditions varying, where we have relatively lower productivity in the oceans, higher productivity, and then some mixed productivity. So we have all these natural things going on that are varying production, but we also have other things that is occurring in recent years. This is a figure of recruits per spawner over time. Recruits per spawner, you can think of that as the number of babies produced per mom. And you can see that it tends to vary historically between about 4 and 8, bouncing around. And then in recent times, there was this decline in recruits per spawners, getting to the point where we're below 1 in some recent years. So these populations are not replacing themselves. And it reached the all-time low in 2009. Now, during this period of declining salmon abundance and declining recruits per spawner, there have been many issues going on with the fishery. And in some cases, very confrontational issues. And in this case, this is a picture from 2005 where there was a commercial fishery protest occurring because the fishermen weren't getting enough of what they thought they deserved given the number of fish that were returning that particular year. We have problems like that going on all the time. However, in 2009 we had a particular issue that reached national prominence, the Globe and Mail, where we had apparently 10 million salmon expecting to return, and we only had 1.7 million returning. So where did all the fish go? This record low return, along with that general decline in productivity, led the federal government to create the Cone Commission of Inquiry into the Decline of Sockeye Salmon. So this federal inquiry cost millions of dollars, and it ran for 2 to 3 years, examining the science management policy issues that might contribute to these patterns of decline in productivity and spawning abundance. It was a very controversial, contentious adversarial process. Some of my colleagues in the room, myself, were involved in it. It was not very pleasant. Did you enjoy your time there, Andrew?
Were you on the stand with the judge?
I was on the stand for 2 days with 20 lawyers, and let's say it was not the funnest part of my life. It was the least fun thing I've ever done. This commission resulted in 75 recommendations. To this date, I don't believe any of them have been formally implemented. But this is a complex situation because during the middle of that commission, we had one of the record high returns ever. Close to 30 million adults returned. Returns best in nearly a century. I think maybe the 4th highest. I can't recall the details, but it was a very large return in 2010. We are now seeing the results of that large return this year with an estimated 20 million fish sockeye returning. Now, we go back and look at the recruits per spawner. Since that period, you can see that, well, things have changed a little bit. We're now back up to somewhat closer to historical levels. So is productivity still in decline? It's hard to say. Turn our attention now to the bigger picture, breaking it down into groups. So there's about 150 stocks, or I like to think of them as metapopulations, within the Fraser system. They have very different characteristics in terms of where they go and how they get there. So the migration distances vary from close to the ocean to very far away. Some of these populations are very small, some are very large, and their run timing. Some start migrating upriver in early July, and others not until early October, and then everything in between. So we see a range in this. A lot of the work of my group has gone into understanding the differences within those groups in terms of these characteristics: body morphology, heart morphology, energy density, fecundity, egg size, and performance of these fish physiologically. And what we found is that the differences among groups of fish seem to be related to some level of adaptation that they need for completing river migration and spawning. Some of the work has been done by my colleague Tony Farrell and his lab, and we've worked together quite a bit in recent years on this. This is a swim tunnel that's been dragged to the riverside, and the swim tunnel pushes water through this closed system at up to 2.5 meters per second, and fish are put into the tube. Up here, you get half into the fish here, in the tube, and they're being forced to swim under these conditions in a closed system, and we're measuring, in this case, oxygen depletion from the water at a given temperature at a given water velocity. You can do the same thing also with probes on the fish, and you can measure cardiac performance, and we've done that as well. So the sorts of data that you get from these types of relationships or these types of experiments is you can look at how oxygen is consumed relative to temperature, and you can develop this routine curve, which is how much oxygen they would use when they're at rest. Then you can also develop what the maximum amount of oxygen would be when they're fully swimming aerobically in this apparatus. And the difference between these two things is called aerobic scope. Many of you may be familiar with this concept, but if you're not, it basically tells you the oxygen available for activities other than the routine. But what's important in this is it's temperature dependent. And you'll have a place where scope is maximum, a place where scope is zero. And the place where scope is zero is where fish are dead. So you'll have a temperature where you have maximum scope and a temperature where fish are dead. So Erica Eliason in some of her PhD work did a lot of swimming tunnel work. Many of you may remember Erica. She's currently a postdoc in our group. And she looked at how aerobic scope varied among populations. So what we plotted here is the relationship between aerobic scope maximum versus the distance that those fish would have traveled had they been allowed to go to their spawning areas. What you see is this relationship between distance and scope, where the maximum scope is greater, the more difficult difficult migrations in the red areas and those that have more easy migrations in the blue, you can see differences like this that relate to other things besides how they perform, how they swim. You can see that they have small, more torpedo-shaped bodies. They tend to have much higher body energy when they enter the river from the ocean. These fish are not feeding during the migration. They've stopped feeding 200, 300 kilometers from the Fraser mouth. The ones that go long distances have very few and small legs. They have very high maximum swimming speeds. They tend to have large and more efficient hearts. And as I just mentioned, they have very large and they have broad metabolic scopes as well. So these fish are built for high performance and energy conservation. So we think they're certainly seem to be adapted to deal with the hardships of migrating long distances. And a lot of those populations that migrate long distances are also migrating in the heat of the summer when temperatures are quite warm. So now I'm just going to change gears a little bit now that I've introduced the system to you and talk about this one phenomenon of early migrating adults. Now, for harvest allocation and management, these stocks tend to be combined and lumped together into aggregations based on their run timing. And the run timing can be broken into these four groups: the early Stewart, early summer, summer, and late summer. Now, right now, at this moment in time, the late summers are now just about, well, some are migrating in river. They shouldn't be, but some are migrating in river, and the summer runs are done. So this is where we are in the whole timing of this. The late summer behavior, though, is quite different. Most of these other runs, the early summer, early Stewart, and summers, they migrate out of the open ocean, they hit the coastal areas, they migrate down the coast, and they come right into the river. There's very little holding or delay or milling in the Strait of Georgia or in the estuary area. However, historically, the late summers had a period of holding, of milling. We're not sure of how many other sockeye groups anywhere do this. We haven't been able to define that, but certainly this was a very interesting behavior that seemed to persist and exist for a long period of time, as long as we had data on this. So historically, they held in the estuary. From 1995 to the present, though, segments of all these late run groups have eliminated this behavior, and they're migrating upriver 2 to 6 weeks earlier. Now, not all the fish in every stock are doing this, but segments of them are. To try to get a better idea of this, this is the relationship between sort of the initial upriver migration date for a variety of stocks. These are 4 of the late run stocks that we can consider over time. And you can see that generally, it's about right now, September 12th, when we start to see historically these runs migrating upriver, and of course much later for some populations. The cultus population used to not come in until the 20s. I studied the cultus population back in the early 90s. I was doing telemetry on it. That was when you were allowed to touch this population, when they weren't considered endangered. And I remember being out there tracking them in the snow. Okay, I was in the river, snow coming down in the Chilliwack, and tracking them. Not so much today. So what you see now is a shift in behavior with these groups coming in much earlier, initiating their migrations much earlier. So if we turn our attention now to one stock we've really studied a lot, because we were able to. It's a closed stock, and it has a spawning channel with a gate that we can very efficiently assess numbers of fish returning. We can take a look then at the migration phenomenon and mortality in relation to when they come into the river. And what you can see here on this axis is the upriver migration date. This is assessed as 50% of the run that's past the town of Mission. At Mission, there is a hydroacoustic facility, these side scanning sonar systems that give us indications of when fish are moving by, and then biological sampling accompanies that with using DNA sampling to identify what the stock groups are that are coming by. This is all done by the management agencies. When you go back to the beginning of the data set, you can see that early on in this period, basically the fish were coming in, as I said, late September to October, and very little mortality during the run. However, since then, you have a phenomenon like this, where you have much earlier migration of much of the run into the system, and occurring for all years, and associated with that is much higher mortality. In some years, almost complete extinction of that particular year class. And this is a lot of fish. It's not just a few fish, but in this group, although it's not a big population, beaver, in some years that was a lot of fish that were part of that mortality figure. So this is what I'm calling early migrants as being dead fish swimming. They get into the river, they head to where they need to go, but they don't get there. Some fish do get there, but they don't spawn. So we know that Pacific salmon are semiparous. They're going to get to the spawning grounds, they're going to spawn, they're going to die. Whether they spawn or not, once they're on the spawning grounds, they are going to perish. The question is, do they get to spawn or not? What we see with these late run groups, again, at about that same time period, is you see pre-spawn mortality. That is arrival to the spawning ground, but dying before spawning, also increasing. So you may be able to make it into the river and make it to the spawning grounds, but doesn't mean you're going to necessarily spawn. So whatever is going on with these late run fish, it's affecting them during the migration and also affecting them on the spawning grounds. Is it happening today? Well, it's hard to know because today is the 12th. This is when fish are supposed to be coming in. Based on some in-season estimates, there should be about 10 million late runs, we believe, that are going to make their appearance and migrate upriver. We've already had 1.4 million migrate upriver. So yes, early migration has happened again to some degree. It's hard to tell how big of an issue this is this year because 3.3 million to date have been captured. Some of that has been in river, so they were early migrating if they came in the river or caught, but most of this capture occurs in the coastal areas through large purse seine fisheries. So we don't know if those fish would have been an early migrator or not. Perhaps one of the better indicators of this phenomenon is the Harrison River sockeye. Harrison River sockeye generally are one of the later ones to come in. What we found this year, and every year, is the peak of the run in Harrison sockeye was August 15th. So 500,000 fish passed mission by August 15th. In recent years, this late run stock is now being called the summer run stock. So you can maybe get rid of the problem by just renaming your population. I don't know. All right, so what are some of the consequences of this? Well, certainly total number of late runs starting the migrations varies quite a bit from year to year, maybe 1.5 million on average per year. But what it means is that since '96, tens of millions have started the migration upriver and have not reached spawning grounds or have reached them but not spawned. So what does this mean in terms of fisheries? Well, in recent years, fisheries have been significantly curtailed or stopped on late run stocks. Not this year, it's a fairly large run, so they could perhaps sustain a much higher exploitation rate, even with early migration-related mortality. But it also means that some of those smaller stocks, like cultus, are on the verge of extinction all the time. They're very small, and they're always at risk because of this early migration issue. So in 2002, my colleagues and I began a large integrated research program to investigate why are they migrating early, why are they dying. And what was bizarre at the time is we had no baseline to compare it to. We had no understanding of the basic behavioral physiology of adult salmon migrations. Nobody really did. So here I'm going to acknowledge my co-authors in this research program and also in this review paper that reviews all this is Steve Cook, Tony Farrell, Christy Miller, Michael Point, and Dave Patterson. And their groups and my group came together along with Carl English and Dave Welch and some other consultants to really spearhead some of the work into this. The study methods involved lots of destructive sampling, so bumping and grinding fish, getting them wherever we could, looking at what was different between fish at different points in their migration and different stocks. We also did a lot of radio and acoustic tracking of fish, and we continue that today, and many swimming performance and telemetry experiments, and I'll review a little bit of all that in the next few minutes. So the types of transmitters that we used include acoustics and radios. The main difference, if you're not familiar, you can see there's no antenna here. This uses sound. This uses radio. So you need an antenna. This is a particular transmitter that has sensing electrodes that would go into muscle tissue. Most of ours don't use this type of transmitter. You can see how big they are relative to a dime. And we attach these eye-button thermal loggers to each transmitter we put in because there's a chance we're going to get that fish back from a fishery or from the spawning grounds, and it has remarkable information that carries with it then on its thermal experience, which is important, and I'll talk about a little later. So we have acoustic receivers and radio receivers. We have acoustic curtains that get placed out into the marine environment. Here is a big messy map that shows what the configuration of some of our receiver stations and tagging areas looked like just a few years ago. It's a little different today, but not a lot different. So we would have tagging sites up on the coast. We have some closer to home here and here. We had tagging sites in the lower Fraser and some up in the Thompson River. We had receiver curtains, and still some of these are here today. These used to be former post receiver curtains. They're now, the ones that are still in existence are called the Ocean Tracking Network Canada receivers. So there and there, if you come into the Fraser, we had one at this about 6 years ago, that went around the mouth of the Fraser completely. And you can see receivers, systems, radio, and acoustics spread throughout the watershed. Our focus of study was these dots, the populations going to the Adam Shuswap, the Portage, the Harrison, Weaver, Cultus. Those were the late run stocks of focus. When we're doing our tagging in the ocean, we charter commercial purse seiners to help us get them. They collect them using their net, and we braille them out, and then we would use individuals. Usually we braille fewer than this, so we're as gentle as we can when we're getting these fish, because we're relying on them to take transmitters and also do other things to them. The other things that we do to almost all of our fish is we take blood from them to examine plasma ions, osmoregulatory, reproductive stress hormones. We, in some cases, take a little bit of gill tissue, just a little filament, off of the gill arch to look at ionoregulatory function and some genomics work. Sometimes we take muscle plugs. We always take some adipose tissue so that we can look at DNA stock assessment, because we have to know what is the stock of these fish, where are they supposed to be heading. We need that information after the season to analyze the data. We sometimes take gross somatic energy using a microwave energy meter, and transmitters almost always go down the throat quickly. We do insert some surgically. That's only if the fish are still feeding, so if they are a long way from the Fraser River mouth. But in most cases, they go into the stomach. Remember, the stomach's not being needed. They're not eating. The stomach is shrinking. It's actually an ideal place to put a transmitter, because the stomach actually just closes around the transmitter and holds onto it for us. So it's a great place to put a transmitter, and the whole procedure can be done in just a matter of 1 or 2 minutes, and then the fishes are just released. So I'm going to show you a few figures that look like this. Answer the question, why are they leaving the estuary early? This particular one will deal with osmoregulation. So here a fish would be captured and tagged and released at the X. They'd be crossing over one of the receiver curtains that were here, and there's a receiver curtain here, so they would have the decision. They could hold like they used to hold, or they could come into the river directly and not hold, and they would be picked up by receivers into the river. In this particular figure, I'm showing you results of gill sodium potassium ATPase. So it's an enzyme that is related to ion activity, and you'd expect the activity to be quite high when the fish is in the marine environment. And if the fish were out in the open ocean swimming around, you'd expect it to have really high levels like this. Well, these fish that we're not holding or migrating in are behaving like full saltwater fish. The fish that we're holding are doing what they probably should be doing. Their levels are dropping to be very similar to what a freshwater fish would have. So very surprising result. These fish were coming in. We anticipated it might be the other way around, that fish would come in early because they think they're a freshwater fish, but that's not what we found. Here's a similar type of figure, in this case looking at gross somatic energy measured from the energy meter information that we have. And you can see the fish that aren't holding had much lower energy than those that were holding. Now these energy levels of no holding is very similar to what you see of a fish much further along the river migration. And finally, some results from reproductive hormones. This is just one of several we've looked at. This is female testosterone. And you can see, you can see that with the fish that we're not holding, significantly higher, really high testosterone levels. These are levels that we see as the fish was migrating well upriver, approaching spawning grounds, compared to what they were when they're holding in the estuary. Again, suggestive that being more sexually mature might be one of the cues pushing them into the river quicker. So if we just summarize some of these things, and I'm only showing you yes and no results here, but as you can see, we've looked at this many times. Steve Cook and Glenn Crossan did lots of work on different years, different populations, and this is a repeatable pattern, it seems, that we're not seeing any premature changes to the osmoregulatory system. They're not becoming freshwater prepared earlier, and that's what's coming in. If anything, it's opposite. Do they have depressed energy states? Yes. Do they have advanced reproductive states? Yes. And those two things probably work together because they're probably preparing their reproductive organs and diverting energy away from somatic reserves into gonads, which is probably why they have much lower lower reserve energy. I didn't show you any data on enhanced physiological stress. We had lots of measures for that. We didn't see any relationship there. So the implications of this is that early river entry fish are motivated for freshwater entry in terms of the reproductive levels, but they're not physiologically prepared for this. And that's certainly clear from the fact that their ion exchange systems aren't prepared. Is this a contributor to the mortality per se, or is this just correlated with the behavior? And that's something we're not still clear on, but I'll show you some results towards that end in a moment. One thing that was clear is the genesis of this phenomenon is not the adult coastal migration. It didn't just happen a week or two prior to them getting to the mouth, because those reproductive hormone levels, for them to be precipitated to change that quickly to that level, would have had to happen much earlier in their life history, probably when they were off the coastal shelf in the open ocean. So something earlier in their life, whether in the open ocean to the river. They get into the river, what happens to them? So here is a historical thermograph, temperature related to month. This is daily averages over this time period. This is the average. This is plus or minus the confidence interval. And you can see that the runtime meters differ in their thermal experience. The lakes, historically, would have encountered about 13 to 14 degrees when they came into the river. Things have changed. So, we just go back here. The lakes are now migrating over here, and the temperatures in the river have warmed by at least 1.5 degrees, almost 2 degrees in some cases. So we now have temperatures the lakes are encountering that are 5 to 6 degrees warmer than they historically encountered. So you can imagine, in almost a turning of a switch, these fish are now encountering temperatures 5 to 6 degrees warmer than they historically used to deal with. Is that a big deal? Should we be concerned with that? I'll come to that specific aspect in just a moment. First, I want to show you that we do lots of work at our field lab in the Fraser Valley at Cultus Lake. We bring salmon there. We're actually at this very moment offloading salmon from the ocean to DFO West Van dock, and we're driving them to Cultus Lake at this moment to do experiments. This is an example of some of the results of some of those experiments that we conducted there. In this case, we're looking at mortality rates, mortality percentages versus time being held in tanks at different temperatures. This is sort of a summary schematic of the things that we found there. So you can imagine this is how long it would take for them to migrate upriver to get to spawning grounds. So by the time they get to this gray area, these fish should be more or less reaching their spawning areas. So natural senescence and mortality you'd expect to start to see out here. So what happens in here is going to have a lot to do with the temperature that we're giving them. So when we hold them at cool temperatures, what they historically may have experienced, there's really no effect on survivorship. In some cases, we cooled the tanks, we warmed them a little after that time period. Didn't really matter what we did in that effect, nothing changed. However, when we started to warm the temperatures before they got to their presumed spawning ground timing, we started to see mortality rates pick up a little bit. If you warm them just a few degrees more, you see a dramatic change in survivorship with over half of the fish dying in just over a week. And it didn't matter if you cooled it a little bit, they still continued that trajectory. What was interesting, that was a bit of a fluke when we did it, is we cooled one of the tanks and we saw, well, if you cooled it a lot on this trajectory, you can actually slow the mortality rate somewhat. And this is actually really important because it was our first clue of what the importance of thermal refuges could mean to these fish. And I'll come back to that also in a few minutes. So there are lots of different causes of thermal-based mortality. You can sort of think of these as either fast processes or slow ones. The fast processes could be mortality. So back to our lovely figure here of aerobic scope, just to remind you how it's calculated. So we are now going to show you the relationship between aerobic scope and temperature for 3 groups of salmon. They're not all sockeye. So this is a fitted curve to the aerobic scope data that we would have collected using our respirometers for the Gates Creek sockeye, the Beaver Creek sockeye, which is the summer run, a lake run, and Chehalis coho, which is much later in the fall. And the dotted lines are extrapolations to the axis. It's difficult to keep fish alive at high temperatures, it turns out. And so we had trouble doing experiments with these higher temperatures, so we extrapolate that down. And those temperatures would be the T-crit temperatures. So what we see when we look at these relationships is that, first of all, the absolute aerobic scope and estimated T-crit varies among populations. Now, I showed you some information earlier from Eric Eliasson that showed that as well. But what's really interesting is that there's not a lot of difference between their best temperature and their worst temperature. It's only about 6 to 7 degrees difference between when they are performing maximally and when they're dead. Okay? Doesn't matter what population you look at. So that's an interesting finding. Then we take those same curves and we overlay on top of that their temperature experience. So this is the temperature frequency distribution of what they historically would have received in terms of their thermal experience over many years. And we plotted it underneath each of these curves. And what you can see is a pretty good concordance between where the peaks occur in their temperature experience and their optimum swimming performance. So the key point here is that their thermal optimum seems to be coincident with the historic river temperature profile, and this is suggestive of thermal adaptation, that they are doing best at the temperatures that they tend to experience most frequently. Okay, so I'm going to go away from metabolic scope and turn my attention to more longer-term processes like bacterial issue. We have fungal infections, saprolinea. I'm sure you've seen many furry fish on spawning grounds. If you've gone to look for them, that's usually saprolinea. These things happen largely because they start losing their protective slime, at least the fungal infections. And so they're opening themselves up to infections of fungal nature. Here's a good example of Columbus. You can see the bacterial infection that occurs around the gills. We've also spent quite a bit of time looking at pathogens in this particular of parasites. In this particular case, this parasite is called Carbocapsula minibicornis. It's a myxosporian parasite that occurs naturally in the Fraser estuary. It's picked up by all adults when they migrate through. On this axis, we're looking at severity of infection of fish taken to the lab and held under varying accumulated degree days, so being held at different temperatures for different lengths of time. And what we're looking at is this maximum score of this infection in the kidney. And so you can see that there is a relationship here with infection in these lab-held weaver fish increasing rapidly after about 370 degree days. Well, so we know that they get sick, but what does that mean to survivorship? In a separate experiment, we had fish, same group of fish, taken to the lab, held for about 3 weeks at 18 degrees, and we start seeing mortality start to begin at around 300 degree days, and then it ramps right up. And if you overlay the last figure on top of this one, you see a pretty good concordance. So suggesting that there's some link between the disease progression and the mortality, at least in these lab-held fish. It didn't really answer the question, though, is temperature causing the problem? So we did an experiment to test that. We went to the field, and we took fish that were on the route to Weaver Creek, and they were still 3-4 weeks away from spawning period. We captured them, we brought them to our lab, we treated them there to 1 of 2 temperature treatments. Either they were a cold treatment where they had over 300 degree days, or a warm treatment where they accumulated over 500 degree days. We took those fish, put transmitters in them, and made them swim it again. So we do this quite often. And so these fish are migrating back up to their spawning areas. I'm going to show you a figure in a moment where we're just looking at survivorship from the confluence of the Fraser and Harrison River to their spawning grounds, a mere 8 kilometers. So here we're looking now at mortality as a percent of the groups we've released over the 8 kilometers that we've released that we're tracking. And what you see is that the cold-treated fish, both groups, suffered some mortality as a result of the transport, releasing them and letting them track themselves back up to that point, but it didn't differ between the temperature treatments. But what was interesting is that in that very short distance, we started to see a divergence between these 2 groups, with very little change in the cold treatment, with the warm treatment over doubling in terms of the mortality rates in that 8-kilometer section. And in fact, when you go back to that year and take a look at it, the mortality rates actually reflected what we had there in the early time migrating group and more with the normal time migrating group. So in some ways, we have reflected what we saw in nature, and certainly we're able to show that temperature was the cause of the higher mortality that we're seeing. Now I mentioned those little eye buttons that we stick on fish that we get really good thermal information from. And if we get the fish back, we can retrace the path that the fish took, or certainly its thermal history, and think about the path that the fish took. So here's some information for one fish that was tagged out here in Johnson Strait, and it followed this migration trajectory. You're going to see a series of colors now on this axis and here that are going to match up. So fish is in the ocean, the Fraser River, the Thompson River, Kamloops Lake, and so on. These colors here match these colors. So the fish is in the ocean, and this is the sort of thermal experience it has. You can see that in some cases, some days it's quite hot. Some days it's quite low. And so we have this variable thermal experience. And what we think is happening is the fish is actually physically going up and down in the water column. But at the same time, you also have, in some days, warm water coming off the Fraser that's spilling out, keeping the surface of the strait quite warm. So part of the warming is because it's encountering that warmer surface temperatures, but it's also going clearly down into the deep areas as well. However, once it leaves the ocean, it hits the Fraser River, it has no choice but to accept the high temperatures that it gives it. There's no thermal refuges in the Fraser River itself. So what it's doing here is it's tracking the temperature of the river as it migrates upriver. Then it hits the Thompson River, and experiencing the Thompson River temperatures. Then it hits Kamloops Lake. What does it do? It goes down deep in the lake and traverses the lake at depth. So it's getting some form of system, and then into Shuswap Lake. Does the same thing. It migrates through mostly in the deep portion of the lake, comes out of the other end, and into the warm Adams River. So, we've looked at this now in many different systems, many different populations, and this behavior is consistent. When they transit through a lake, they do it at depth. When they're doing it for clear reasons to get a thermal refuge. We've also examined fish that don't do it versus those that do it if they're early migrants. And the early migrants that do it survive better than the early migrants that don't. Just about 510 degrees. Okay, so the take-home temperature. So take-home temperature. Yeah, temperature. Take-home message. Warm temperature plays a key role in mortality of early migrating lakes, and the critical temperature limits, like metabolic issues, and accumulated degree day limits, like disease issues, can explain much of the migration mortality, but there is still more to the story. And I think I've found it, right? Oh, perfect. So, capturing and tagging lake runs about 500 kilometers upriver. We did this with some First Nations. We were using a dip net, which is a very gentle way to get these fish. And what we found is when we took them out by dip net and sampled their plasma lactate, which is an indicator of metabolic stress, we found that the fish that were normal time, those that died on route to spawning grounds from that point forward and those that didn't die, there was no difference in their metabolic stress. However, there was a large difference in those fish that were early time, suggesting that the early fish did have this evidence of metabolic stress. There was something else going on with these fish. We weren't often cutting them up during the migration, but in this one study, we were attempting to insert electromyogram transmitters, which had to make a little incision in the stomach, in the abdominal cavity, I should say. We found excessive bleeding with the early time migrants, and this was very odd because we'd never seen this before. So what we did is we looked at blood clotting, time to blood clot using blood tape from these fish, and the early fish took twice as long to clot as the later fish. So there's something going on with their blood and their clotting. Is this symptomatic of a larger problem? Is there a larger problem? Well, to look at that larger problem, we started doing functional genomic work with Christy Miller. And if you're not familiar with functional genomics, these are, in this case, we're using microarrays, which are microscope slides that have printed on their surface gene transcripts from the species. Now, you're basically looking at the genetic control at the RNA level, looking at the physiological processes that are involved. The microarray, this particular microarray, contains over 40,000 gene spots, so it spans most of the genome. What we found was that in these ocean tag fish, a mortality-related genomic syndrome. It was associated with a 13.5-fold greater chance of dying en route. And that involved about 1,000 genes. And when you interpreted these genes, it came up with 3 issues. One was a blood issue, in terms of downregulating the clotting factor. Second was a metabolic issue, in terms of downregulating fatty acid metabolism and aerobic respiration ability. And third had to do with disease and stress. There was a problem with immunosuppression. Taken together, this is supporting everything that we've shown with a lot of our other biomarker work, but when Christy was interpreting this, it looked like it was the signature of a viral infection. And this viral infection turns out to be a novel virus that we hadn't seen before, that appears to be similar to a flu-like virus. The take-home message is that early migrating lake runs are in poor or altered physiological state at the start of the migration, and then the high river temperatures makes it even worse for them. Okay, lastly, and this is just a couple of slides, so I should finish on time, is if we take it back to the big picture. I focused a lot on lake runs. Let's look at all the stocks now. And here, what we're looking at here is all of the big stocks across their stock river entry timing. And here are their run timing groups, or these stewards right to the lakes. And what you're looking at here is the number of years when mortality was greater than 50%. So we're looking at big mortality years. How many do those exist for each group of fish? What you find is that 9 of the 12 stocks had more than 50% of their recent years of high mortality. But what was really interesting to us is that there were stock specific, some didn't have any high mortality years. Some had really high mortality years. And it seems to be the ones at the ends that were exhibiting this. If we take this back to aerobic scope, so here is the percentage of maximum aerobic scope versus temperature. And we're looking at now 6 different populations here. And we just contrast the 2 extremes on that figure, which are Weaver and Chilko. The Weaver population here has its T-crit at just over 20 degrees. It runs out of scope really quickly. The Chilko fish don't run out of scope quickly. In fact, they have lots of scope to spare all the way up to 28, 29 degrees. That's a remarkable difference within 2 populations of fish that only migrate about 3 or 4 weeks apart from each other coming into the river. And show you that this temperature difference occurs also when we're looking at our telemetry data. So this is, we're looking at now over 1,500 fish carrying transmitters over a 5-year period. We're looking at modeled average survival of these fish tagged in the ocean and survivorship assessed in freshwater. We see with choco, very little effect of temperature across the range of temperatures that they experience that we study. But we start to see changes at higher temperatures in these other populations. For most stocks, 18 degrees is the tipping point, but this thermal migration is certainly stock specific. And just to add one more wrinkle to this picture, if we grab the fish in fresh water and tag them and release them, these are the results. What it means is that remember, these fish were tagged in the ocean and then tracked once they got into freshwater. These are fish that were tagged in freshwater and tracked in freshwater. In each case, the fish tagged in freshwater and tracked in freshwater didn't survive as well. But you still see stock-specific differences. In some cases, the differences are quite stark. But perhaps most troubling is the fact that when you handle a fish in freshwater and release it, there's these strong latent effects if they encounter high temperatures. And so all sorts of bells and alarms should be going off thinking about in-river fisheries and what does this mean for fish that are captured and released and they encounter high temperatures. Not good if you're a true tropical fish. So survival of freshwater caught with these fish poor warm temperatures, especially for the lake road. So what's the future hold? Well, the river's going to continue to warm. I think we're all in agreement that's going to happen. Another 2 degrees. We'll continue to see high migration mortality for late runs if this early migration continues. Certainly those early migrants aren't surviving. That's bad news for weaver fish. Other early time stocks will probably also be affected. Remember, it seemed to be the early time fish and the late time fish don't have the ability to swim really well in high temperatures. But these summer runs may be okay. Shellfish we dug in superfish. They seem to be doing really well regardless of the temperatures that they're currently encountering. They have lots of scope left to deal with higher temperatures. We do expect pathogens and disease to figure prominently as the mortality agent as the Fraser warms, and the potential exists for salmon to cope with a warming Fraser. It's not all doom and gloom. Physiological adaptation can occur, and it can occur sometimes rapidly. And we can see phenological changes like altered river entry timings to cope. We've certainly seen this in the Columbia with certain stocks changing their river entry timing to avoid high temperatures. We haven't seen that yet in the Fraser. There's little evidence to support that this is happening, but it could. So, which I'd like to thank lots of supporting organizations, lots of agencies, and lots of colleagues. I certainly want to thank all my lab members. This is an old photo. I looked yesterday for a much newer photo. But there have been dozens and dozens of grad students and postdocs and research associates that have done this work, and I'm just the one lucky enough to present it all. So with that, I'll end. Thank you.
So, this is Carl.
Yeah.
Right now, the peak of the Quesnel run is just out here in Quesnel Lake, and according to the temp depth you showed migration at, we've got people looking, and they are down there in the summer climate. They're encountering really strong gradient in toxic metals and stuff from the Mount Pollard thing. They're only going to be in that water for about a day and a half. If they refuse to enter, and they do avoid copper in the water, especially, what do you predict the impact would be of 2 extra days for them of having to stay up in the lake surface water before they can get to clean water up the lake?
Well, so the summer runs probably have a better ability to deal with warmer temperatures than a lot of the late run data I showed you. So being a summer run, I would think that a couple of extra days wouldn't have a huge effect because we're only talking about 40 or 50 degree days.
Yeah,
So it's that by itself is probably not going to be what hurts them.
yeah, well, that's good news.
Yeah.
The chemicals aren't concentrated up to the top.
Yeah. I mean, the bigger issues with this depth sounding, and this is the case in Okanagan and the Soyuz Lakes, but the Okanagan run is the anoxia that can occur. So they want to seek thermal refuge, but it's a very narrow band where they can get it.
Right.
And then a lot of what goes on in those lakes is recreational fishing, where they pull the fish up, I don't want it, put it back, and so they then experience these really high temperatures as well as the oxygen limitation down there. So you push the oxygen issues even further by doing that. changing early life growth, and how that might affect growth patterns and subsequent maturation rates could be part of that. Food quality, which we didn't study at all in the ocean, could also be part of that, and how it affects the rates at which and where they start to turn. It has a lot to do with light levels and food and growth potential in the ocean when they decide, I'm going to mature and come home. And those are things we just couldn't study because we're not out there. So we haven't identified why. We have lots of correlates. I think we've identified what happens when and what factors. Yes.
The thermal, the differences in thermal ranges, do you have any idea of whether those are just selected for, or whether it's some sort of physiological plasticity that's set through early life experience?
Yeah, that's a really good question. And the only way to test that is really to look at them when they're little. And so Tony Farrell and colleagues have started to do that with some other species of salmon, and I don't know what they're found yet. In our experience, we haven't done the swimming performance. What we've done is we've looked at eggs and fry survivorship under different temperatures, and interestingly, they do best under the conditions that you would expect them to do best under, given where they come from. But that doesn't necessarily. So weaver fish are actually really good swimmers when they're juveniles, and they're the worst swimmers when